Hello, College Biology. This is Nucleotides Part 2. In this video, we're going to go a little deeper into the mysteries of the nucleotide. So let's get to it. We talked about how DNA, in the last video, we talked about how nucleic acids are the instructions for building proteins, and the polymers are DNA and RNA, and those polymers are made of nucleotides, which we have here, and those nucleotides have different parts, and every nucleotide has 3 parts, and those are the nitrogenous base, the sugar, and the phosphate. And the phosphate is the same for all nucleotides. It's just a phosphate group. However, there can be variation in the nitrogenous bases, and there can be variation in the sugars. And when we take the nitrogenous base and the sugar and put them together, that's a nucleoside. We finally add that phosphate, and now we have a nucleotide. So we are going to talk about the variations that we can have in our nucleotides based on the nitrogenous bases and sugars. So let's go to our nitrogenous bases. First, nitrogenous bases can be divided into 2 categories. Those categories are pyrimidines and purines. And regardless of whether it's a pyrimidine and purine, all nitrogenous bases, as the name suggests, feature nitrogen. And so what they all have in common is that they are a ringed structure that is composed of carbon and nitrogen. So here, carbon and nitrogen in the ring, carbon and nitrogen in the ring. Here, carbon and nitrogen in the rings, carbon and nitrogen in the rings. So the difference between pyrimidines and purines is that pyrimidines are a single ring structure made of carbon and nitrogen, and purines are a double ringed structure made of carbon and nitrogen. As you can see, this is a 6-membered ring and a 5-membered ring, whereas the pyrimidines are all 6-membered rings. So when we say that there's a nitrogenous base, it could be any of these 5. And so there's a variation. Not all nucleotides are the same because they don't all have the same base. And that's good because we can now have variation in our polypeptides. We could make this nucleotide could have a nitrogenous base that's cytosine, and this nucleotide could have a nitrogenous base that's adenine, and this nucleotide could have a nitrogenous base that's guanine. And so when we talk about sequencing your DNA, what we're really talking about is checking the order of the nitrogenous bases in your nucleotides. That's what a DNA sequence is. It's the order of the nitrogenous bases. So if you ever see, and they talk about DNA being spelled in letters, well, these letters, A, C, T, and G, are referring to the nitrogenous bases on the nucleotides in the polymer of DNA. So that's one way that nucleotides can change. And the thing is that these nitrogenous bases, they can form hydrogen bonds with each other. Pyrimidines can form hydrogen bonds with purines, and that is how the cell reads the DNA in order to make mRNA, or how it reads the RNA in order to make protein. So those hydrogen bonds are really important, and we're going to explain and talk more about those in our second quarter. So those are our nitrogenous bases. Our sugars. There are 2 different types of sugar that can be found in nucleotides. Those sugars are ribose, which is a 5-carbon sugar, and deoxyribose, which is also a 5-carbon sugar. Now, ribose is used only in RNA nucleotides, so you will never find this sugar in DNA. So ribose is a 5-carbon sugar that is used in RNA nucleotides. Deoxyribose is a 5-carbon sugar that is only found in DNA nucleotides. And the name deoxy is meaningful because deoxyribose is actually exactly the same as ribose, only one of the oxygens is missing. So it's not like they deoxified it by taking away all of the oxygens. No, they just took one away and called it deoxyribonucleic acid for DNA when it's in a DNA nucleotide. So that's it for nucleotides. They have 3 parts. The phosphate is always the same. The nitrogenous base can change and be pyrimidines or purines. And the sugar, if we're making DNA, the sugar is deoxyribose. And if we're making RNA, the sugar is ribose. So let's review. Nucleic acids are made of nucleotides, and each nucleotide includes a nitrogenous base. If it is a purine, it will be 2 rings made of carbon and nitrogen. If it's a pyrimidine, it will be 1 ring made of carbon dioxide, and these purines and pyrimidines form hydrogen bonds with each other, which is needed to make proteins. We can read those hydrogen bonds to make proteins, and we'll talk more about that later. Each nucleotide has a sugar. The sugar is ribose. but that's only used in RNA nucleotides. And the other sugar is deoxyribose, which has one fewer oxygen than ribose, but that is only used in DNA nucleotides. And there's one more thing I forgot. I forgot to discuss ATP at all. So let's take a look at that before we come back here. ATP is a modified nucleotide. It is not a monomer. We do not use ATP in our DNA or RNA. It's not. But we start with a nucleotide, and then we can change a nucleotide into ATP. This is our nitrogenous base. Here's our ribose sugar, and then there's a phosphate here, and from here back, this would be our nucleotide. But if you continue to modify ATP with phosphates until you have 3 phosphates, you get adenosine triphosphate. And now this is the primary energy currency of our cell. So we have a short-term energy storage in glucose. When we get the energy out of the glucose, we put it into ATP, and ATP is like spending that as cash. ATP is the dollar bill of energy. It is the energy currency of the cell. So our primary energy currency comes from a modified nucleotide. So let's take a look again. So ATP is a modified nucleotide with 3 phosphates instead of 1, and it is the primary energy currency of the cell. Sorry, I was a little disorganized about that, but that's what we need to know, and I hope this helps. Struggle well.